Good morning students. In my last video we have discussed about the types of cells, in which the important types of cells are two types. One is prokaryotic cell, other is the eukaryotic cell. So we have discussed about the difference between prokaryotic and eukaryotic cell, and I have told you to write down in your notebook about the important differences. I am noting down these 6 differences. There are more differences which you can find from your book, and please learn this. These differences are very important for the exam point of view. So I am giving you recap of these differences. In prokaryotic cell, true nucleus is absent, but in eukaryotic cell, nucleus is present. In prokaryotic cells, nuclear membrane is absent, but in eukaryotic cell, nuclear membrane is present. In prokaryotic cells, Golgi apparatus, endoplasmic reticulum, mitochondria are absent, but in eukaryotic cell, Golgi apparatus, endoplasmic reticulum and mitochondria are present. Prokaryotic cells are unicellular, but eukaryotic cells may be unicellular as well as multicellular. In prokaryotic cell, genetic material which is known as DNA, the genetic material lies inside the cytoplasm, but in eukaryotic cell, the genetic material lies inside the nucleus. Example is blue-green algae, bacteria, mycoplasma. In eukaryotic cell, plant cell and animal cells are there. We are having the structures of plant cell and animal cell, which we are describing at the last after determining the components of plant cell and animal cell, like what is cytoplasm, what is nucleus, what is endoplasmic reticulum, mitochondria, all about this. So in today's topic, we are coming for the structural organization of cell. A cell is made from the important 3 components. Very first component is cell membrane, second component is nucleus, and third component is cytoplasm. So step by step we are explaining all the 3 components. On the today we are discussing about the cell membrane. So very first topic is cell membrane or plasma membrane. It is the outermost covering, like we having the skin. Okay, so skin is known as the cell membrane. So it is the outermost covering that separates the content of cell from the surrounding environment is known as cell membrane. So suppose if any wound gets and our skin gets removed, then you can see the blood comes out. So this interacts with the environment. Okay, so the cell membrane is the covering which separates content of cell from the surrounding is known as cell membrane. What are the functions of this cell membrane? It forms a cell boundary and maintains individuality. For example, if 50 students are present, they are separated by a cell membrane from each other. Okay, so if a large amount of food is there, large amount of chapatis are there, and if I am packing them in the polythenes, so after packing, the 2 polythenes are separated, the 2 chapatis are separated by a polythene layer. This is also known as the cell membrane. This cell membrane protects the cell from injury and contaminants. Contaminant is the mixing of any foreign substance, that is mixing of impurity. For example, if our skin is not there, then the blood gets mixed with soil when we play in the open. Okay, so to separate, to save this contamination and injuries, cell wall, cell membrane is very important. Next, the cell membranes are semi-permeable. It is very important. Semi means half. Permeable means allowed to pass through it. For example, our skin is also semi-permeable. It allows very less particles, very less substances to go inside or to come inside. Like it don't allows blood to come outside, but it allows sweating to come outside. When we play, when we play in hot, when in summer, the sweating comes out from the membrane. This shows that the cell membrane is semi-permeable. And next the topic we are discussing is what is endocytosis. The intake of any material is known as endocytosis. I hope so in my next definitions I am also writing about the endocytosis. So next again we are discussing about this. So cell membrane are semi-permeable. If I am asking fully permeable, complete permeable, that means it allows all the particles to transfer through this membrane is known as completely permeable. Okay. So next is the structure of cell membrane. The cell membrane is about 6 to 10 nanometer. Nanometer is the very smallest unit of measuring. So the size of cell membrane is 6 to 10 nanometer layer composed of phospholipid layers with proteins. So you are seeing here these ball and two lines. This is known as a phospholipid. So there are large number of phospholipids present. So these layer of phospholipids make cell membrane when proteins get sandwiched in between them. Proteins are of two types. One is the intrinsic protein, other is the extrinsic protein. What is the intrinsic protein? If the protein gets completely inside in between the membrane is known as intrinsic protein. Like these 1, 2, 3, 4. These 4 proteins are completely inside the membrane known as intrinsic membrane, intrinsic protein. But you can see 1, this 2, 3, these are the extrinsic proteins which are present on the top of the layer and having some relation with, having some sharing with the atmosphere also. So this is known as the extrinsic protein. Two types of proteins are there, intrinsic protein and extrinsic protein. So in the first chapter of chemistry I have also studied about the diffusion and osmosis. Here again I am discussing about some diffusion. Diffusion is the movement of substance from one region of higher concentration to a region of lower concentration. Okay, so movement from higher concentration to lower concentration is there. So this is the important, and we have studied about the 3 types of diffusion: diffusion in solids, diffusion in liquids and diffusion in gases. So one new term, that is the osmosis, because in diffusion there is no need of any semi-permeable membrane. There is no membrane required, but here cell membrane is present. So we are discussing a new term named as osmosis. Otherwise the whole term is same. It is the movement of water generally solvent particles. It is the movement of water from high region water to low water region concentration. It is the movement from high concentration to lower concentration through a semi-permeable membrane is known as osmosis. It is very important. So I am giving you an example of osmosis. Suppose when you are bathing for a long time, for 1 hour, for 2 hours in summers, you can see your hands, fingers get swells out. Your fingers get swell out than original. So this is the example of osmosis. When you are continuously putting your hand into water, some of the water gets inside, because in the bucket the water concentration is more. So some of the water from bucket gets into the hand, into the finger through the membrane. This is known as the osmosis. Okay, one more example you should take. Take a egg. Take a egg and put down this egg into water solution. After some time, you check that the eggs get swell up. Swell up means it gets larger in size. So this can also be due to the osmosis. Next is the types of solutions. There are two types of solutions, generally hypertonic. If there are two solutions and one solution is having higher concentration, then it is known as the hypertonic solution. One more, hypo. The solution having low concentration is known as hypotonic. So remember always, if two solutions are there, one solution is having high concentration named as hypertonic solution, other solution having low concentration named as hypotonic. One more solution name is there, that is also named as the isotonic solutions. That is known as isotonic solutions. What are isotonic solutions? If two solutions have similar concentration, then they are known as isotonic solutions. Next heading is endocytosis. I am revising again, because in the last presentation I am giving you a definition of endocytosis. The process of intake of material through plasma membrane is known as endocytosis, and the process of removal of material is known as exocytosis. Remember this definition. Exo means exit. Exit means bahar karna. The process of removal is called exocytosis. The process of taking is known as endocytosis. Now next we are discussing about the cell wall. So I am discussing in the previous presentation about the cell membrane. Here I am discussing about cell wall. Cell wall is found outer to the cell membrane. If there is a membrane, if there is a layer present to the cell membrane, then it is known as the cell wall. It is mainly found in plant cells, which is discovered by Robert Hooke in 1665. The cell wall is always absent in animal cell but present in plant cell. The cell wall in plant cell is composed of cellulose. Strong cellulose is there, which provides strength to the cell. It is freely permeable. Freely permeable means it can transverse, it can permits all solutions through it. It helps to maintain the shape of cell, and in roots it helps in the absorption of water and minerals. So this is about our first term of the components of cell, that is the cell membrane and cell wall. Please note down in your notebooks and be tuned for the next video. Thanks. And